Thank you very much. And welcome everybody. Hopefully this will be an interesting run through of my current project and the results that we've gained so far. So my project has revolved around trying to improve our understanding of the epidemiology of strep uberis mastitis and the different modes of transmission that we're seeing in British dairy herds. So we selected strep uberis as the main target for this project because now it has become the most important single pathogen that we're dealing with. So about 1 in 3 of all high somatic cell count cases are caused by strep uberis. About 1 in 4 of all clinical mastitis cases are caused by this one pathogen. Now, we have traditionally thought about strep uberis really as being an environmental pathogen associated with particularly straw yards and generally poor hygiene, whether that's in straw yards or in cubicles. And also across the world we see strep uberis has been quite an important pathogen. A lot of work has been done in New Zealand in grazing-based systems, and there is some good evidence there about management of the risk of strep uberis mastitis in grazing management. But what we've also been seeing over the last 10 to 15 years are an increasing number of one-off reports from small numbers of herds or small studies, which have implied that there's the potential for contagious transmission of strep uberis as well. So this is a contagious transmission primarily through the parlour, through milking equipment, through other vectors during the milking process, moving bacteria from cow to cow, whether that's the cluster itself or milkers' hands, for example. Now, this really runs counter to the traditional orthodoxy that really we've been thinking about mastitis pathogens, basically in two categories, a black and white view of a pathogen has to be either contagious or environmental. And for the last 40, 50 years, we've been putting pathogens into these two convenient categories, the main contagious pathogens originally being strep A-galactine, Disgalacti, and Staph aureus. And the control measures introduced for the 5-point plan in the 60s were very, very successful in dealing with what then was primarily a contagious mastitis problem. So we pretty much eradicated A-galacti from the UK in all but a tiny number of cases. And what we have then seen is both an absolute and a relative shift in the ecology of the bacteria that we see. So the environmental pathogens, as they were thought, E. coli and strep uberis, have then become proportionately much more important because they're not really addressed by those control measures introduced in the 5-point plan. However, over the intervening decades, we've seen more and more evidence that actually this clear discrimination between two categories, contagious and environmental, is probably an unhelpful oversimplification of the true underlying biology. So what we're seeing, rather than a black and white situation, is really different gradations of grey. So we're seeing bacteria such as A-galacti are very, very contagious and have very little ability to survive and replicate in the environment, and the environment has little ability to act as a reservoir. And then at the other extreme, we have E. coli, which is cause very short-term infections within the udder, doesn't really have the ability very well to pass from one cow to another, but there's a limited amount of evidence for it, so it's primarily an environmental pathogen. But then sitting in the middle, we have pathogens such as strep Disgalacti and strep uberis, which, whilst they may be more common in one niche or another, they have significant abilities to be transmitted by both modes. So what underlies this behaviour has got to be related to the genetic sequence of the bacteria, so the strain variation between individuals within the species, to confer the abilities for a bacteria to spread either contagiously or from the environment. So for contagious transmission, what the bacteria needs is the ability to persist for as long as possible within the udder without being removed by the cow's immune response or by antibiotics, so that it has as many milking opportunities to be transmitted through milking events. So the more persistent the bacteria is, the more likely it is to be able to be spread from cow to cow. So this may present itself as persistent cases, poor cure rates, or recurrent cases. And conversely, your environmental pathogens They are very opportunistic. They may cause very florid, very aggressive disease, but they resolve very quickly as well. So quite high cure rates, but when they do occur, they can be very severe. It's the classic environmental pathogen. So what we're interested in fundamentally as vets really is what different control measures are going to be most appropriate, especially if we have to pick control measures to use the limited time and resources that we have available to be most targeted. So what control, what treatment and prognosis information can we get from the strain variation within the species? So the aim of the project really was to try and understand within the UK industry how common is this contagious behaviour that we've had inklings of its existence within the UK for some time, but no real solid estimates. And can we use existing on-farm data to be able to predict the patterns of disease? So can we use monthly milk recordings, or can we use strain information to predict what the mode of transmission on a given farm at a given time is? We were using the original data from the 52 herds which were recruited for the DairyCo mastitis control plan almost 10 years ago now. And those herds were recruited to be fairly representative of the industry, with cases over 35 cases per 100 cows per year. And we requested the farms to submit clinical mastitis samples from all clinical cases, but no subclinical cases. And then alongside that, we had a very complete set of cow and farm level data on management practices, monthly somatic cell counts, all the NMR recording data that you'd expect, and also calving and dry off dates. So with that information and with those samples, those archived samples, we needed to actually understand what the strain variation was. Now, we used a technique which has been developed and very widely used in bacterial research, which is multi-locus sequence typing. Now, the idea behind this technique is that you take a handful of genes. We take 7 genes out of the bacteria's complete genome of about 1,800, 1,900 genes. And these genes are ones which are very highly conserved, so they're very important for key cellular functions, but they also have a certain degree of variation. So we can then use these key elements as a fingerprint for the whole bacteria. So the very first bacteria of strep uberis which was sequenced, each variant of each of the 7 genes was given the first number, was given number 1, and then each subsequent bacteria that was sequenced, when a given gene had a number of mutations which were identified, these were assigned a number, and then you can then build up a library of different allele combinations, each given its own number. And this then gives you a sequence type, which is a sequence of 7 different numbers for each strain. So the results that we had when we sequenced almost 500 isolates were that we saw a huge degree of strain diversity. So we found almost 200 separate sequence types within the 52 herds. But the key thing is that a lot of these sequence types, so 127 of these sequence types, were only found once within the entire data set. So we have this very, very heterogeneous population, but we have a small number of sequence types. These are the ST6, ST22, ST5, 35, 20, 233, and so on. These are much more common within the data set, and the significance of that will become clear in a moment. So when we then looked at the pattern across farms, we wanted to see how many herds had a given number of sequence types. So on the extreme left, you have 2 herds which didn't have any strep uberis cases at all, and at the extreme right, you have the most heavily infected herd, where there were 20 separate sequence types identified. But we have then a bell curve in the middle with between 4 and 9 sequence types being very common in an awful lot of herds. When you look at how common a given sequence type is across the data set, across those 50 herds, we find that 148 of the sequence types were only found on one herd, so they're to some extent herd specific or limited to a very small number of herds. But at the other extreme, we had 2 sequence types which were found in 20 of the 52 herds. So these are very widely distributed sequence types, very abnormal for the whole population. Now that could be regarded as very interesting in itself, but actually what does this potentially then mean? So what we did is we classified the cases that we saw into 2 broad categories. And each farm here represents, each bar on this graph is representing one farm. The blue bars at the bottom of the graph represent sequence types which only occur once in that herd. Now this is our approximation to the environmental pattern, that we'd expect to see with a conventional interpretation of what strep uberis does. The black bars indicate mastitis cases which occur due to sequence types which are found in multiple cows in the herd. So if they're found in multiple cows, then there is the potential that those mastitis cases occurred as the result of transmission from one cow to the next. And significantly, whereas we were expecting to find a handful of herds, 1, 2, maybe 3 herds, which had a dramatic outbreak of contagious mastitis, as had been reported in some previous literature. What we actually found was that this pattern was very, very common across the whole data set. So 60-odd% of the herds actually had some evidence of potential contagious transmission. And for 1/3 of those herds, it was the dominant type of transmission. So there were more potential transmission events than there were assumed environmental transmission events. So this really shifts our understanding as to what the relative importance of these 2 transmission routes may be. And the full research results were then published as an open access paper at the end of last year. So that's available through PubMed or directly through the Journal of Clinical Microbiology for anyone who'd like to read about that in more detail. Now, if we think back to that pie chart of all the strain types that we have, what we found was that 9 strain types caused 40% of those clinical cases, and these were the ones which were obviously our key candidates for contagious transmission. Now, taking that forward, we then looked at how these 9 strain types were related. Now, 8 of these strain types were very closely related, only differing at 1 or 2 of those 7 different key fingerprinting genes. So they form quite a closely related family, and the vast majority of the assumed environmental strain types were very unrelated to either the potentially contagious ones or to each other. So again, this is what we'd expect from a very heterogeneous population in the environment. What we then did was actually look at how these strain types occur in a given farm over time. So what we then did is we took some of the most interesting examples, and we then sequenced the complete genome, and we could then build the more detailed tree, and we could also look at how these are potentially related to each other. So this schematic sort of combines both the time distribution in the horizontal and also the evolution from common ancestors on the vertical scale. So the labels A, B, C, D indicate when they occurred over time, and as you can see that A and B and C and D branched off from common ancestors, and there is a pattern there which is similar to what you'd expect if you were looking at a pathogen which evolves and is then transmitted, and one case is then related to the other. It's not universal, as you can see with case G, but there's a relatively convincing pattern there which indicates you're getting some ongoing transmission occurring. We then looked at the on-farm data that we had, and we were hoping for a very clear relationship with somatic cell counts or with other monthly recorded data parameters, but unfortunately that data wasn't as useful as we hoped, and partly that is because the intervals at which they're collected, 4-weekly intervals, is really not that appropriate for analysing mastitis cases. We don't simply have the granularity of data. We would need many more frequent recordings within that period to be able to track those changes. So whilst the milk recording data wasn't that useful, what was useful was actually looking at the time intervals between cases. So where we had cases of the same strain type occurring close together, we could see that there's a much greater proportion of the PTEs which occur close together than the S's. These are the solitaries, the assumed environmentals. So if we take the red line as our baseline, the blue line indicates how much more common the PTEs are to happen close together. And this is again what we'd expect to see if it's contagious transmission, because it's more likely for a bacteria to be spread quickly, because that bacteria is really up against the clock in terms of its persistence and duration in the udder. So this really adds some supportive evidence that these PTs are possibly real contagious transmissions. Now, that's useful in terms of understanding the disease, but actually it's not that useful if you're trying to diagnose it on a real farm, a new case on a real farm, because sequencing is very slow, it's very expensive, relatively speaking. So we were interested in testing alternative technologies that would allow quick and rapid and cheap diagnosis of these two main groups, what we thought these environmental cases and these potential transmission cases. So this is a set of schematics from another type of software which uses the protein structure of the bacteria. So basically we take a culture of bacteria, we evaporate it with a laser, we accelerate it over a vacuum, and we see a protein fingerprint for those bacteria. This takes about 48 hours to do, and gives you a result very, very quickly. And what it does is allows us to cluster the types of bacteria based on their sequencing data. So we can see that we're getting clusters in red of the PTEs, and they cluster very differently from the solitary cases. And this indicates that potentially we could have the ability to, if we can build up a history of a given farm with a new case, we can assign it to a particular cluster. We can say, right, you're experiencing more potential contagious transmission now. You're at increased risk for this coming period. You should change your management interventions appropriately. Or conversely, you're experiencing lots of environmental clustered cases. It's primarily a hygiene environment issue that you need to be dealing with. So that was from one farm. This is now looking at 4 farms, and what this shows us is that the populations of the PTs, potential transmission events, on the left, and their accompanying environmental solitary cases on the right differ between farms. And this is quite interesting. So we're seeing different ecologies on different farms. So it really indicates that there isn't a sort of one-size-fits-all. We can't necessarily take a mastitis sample from a cow from a completely unknown herd and fit it to a pattern. We really need to understand and develop a picture of what the pattern is on a given herd to be able to fit a new case into one category or another, because they are quite different, because the bacterial strains that we're dealing with are in many cases quite different on different farms. And what that's given us out of this project is an appreciation that actually we can use the MALDI-TOF, the proteomic technology, to develop an accelerated advanced diagnostic tool, and we've been able to leverage additional funding from Innovate UK to develop this in collaboration with Quality Milk Management Services down in Somerset. And what we've also then done is take that whole genome sequence data, that much more in-depth information on all 1,800 or so genes found in the bacteria, and try to do some further work to identify what the underlying biology is that allows the bacteria potentially to transmit in a contagious manner or restricts it to an environmental transmission. And that's work that's probably going to go on way beyond the end of my project, and opens up some really interesting possibilities for the future. I'd like to thank AHDB for the funding and the support all the way through the project. And I'd like to thank my supervisors, particularly Martin Green and Andrew Bradley and James Lee, as well as all the other researchers who've contributed and participated in the research.